Hello, thank you for turning into my poster. I will be talking briefly about the genome-wide association study of anthracnose disease in switchgrass that I have been working on. I will start off by giving a little bit of background information on switchgrass. Switchgrass is a C4 perennial grass that is native to the North American continent. It was designated as a model herbaceous energy crop by the United States Department of Energy in the 1990s. This has ultimately paved the way for research studies to be conducted with the goal to improve the quality and yields of switchgrass. Switchgrass has the potential to be highly productive and suitable for production on marginal lands. Marginal lands are those that are not suitable for growing food crops. This is important because as the world population continues to increase, the demand for food will also increase. Therefore, all of the available prime farmland will be needed to feed this ever-growing human population. Switchgrass requires minimal inputs of water and nutrients in order to be productive, making it a good feedstock for biofuel production on marginal lands. Switchgrass also has a variety of other benefits, such as benefits to the ecosystem, which make it more environmentally friendly bioenergy crop compared to that of the first generation bioenergy crops, such as corn and sorghum. In addition, both corn and sorghum would need to be grown on prime farmland, which would take away from available land that could have otherwise been used for producing food. There are two main distinct ecotypes of switchgrass that are shown in this picture over here, the upland and lowland ecotypes. The upland ecotype typically performs better in northern climates. It's usually shorter and has an earlier heading date compared to that of the lowlands. The lowland ecotype is better adapted to warmer climates, wetter soils, and will perform better in more southern areas of the country. The next portion of our poster that we're going to zoom in on is this anthracnose portion. So one of the major roadblocks to the use of switchgrass in the northeastern United States is a disease known as anthracnose. Switchgrass anthracnose is caused by a novel fungal pathogen, Calotrichum navitas, which has been previously identified and characterized by Crouch et al. In 2009. The major symptom of anthracnose disease in switchgrass is the formation of these lesions on the leaves, as shown here in the picture on the right. This is actually an example of a fairly high degree of disease severity. Due to the wetter climates of the Northeast comparatively to those of the Midwest, anthracnose disease has the potential to cause significant yield reductions, making it a much less economical bioenergy crop. So, anthracnose is the major roadblock to increasing the yields of switchgrass in the Northeast. So, now we're going to zoom in on our goals portion of our poster. So, the two major goals of this study is to first gain a better understanding of anthracnose disease resistance in switchgrass. And then the second goal is to use this information to aid in the development of disease-resistant, high biomass yielding cultivars that are well suited for growth on marginal lands. So now we're going to zoom in on the experimental approach. The association panel of switchgrass used in this study was initially created by Liu et al. In 2013. The association panel contains a total of 540 unique genotypes that have been previously, in which the S&P markers have been previously identified. For this study, the association panel has been replicated across 3 separate field locations, which are corresponding to the dropped pin shown over here in this Google Earth image. You have the Penn State site, which is in Phillipsburg, Pennsylvania, and corresponds to used mine land. The Cornell site is in Ithaca, New York, and that corresponds to prime farmland. And the New Jersey or the Rutgers site is in Freehold, New Jersey, which is also prime land. I've included this map of the United States down here, and this kind of shows you roughly where each ecotype will perform best in which zones. So you have the upland in these top northern regions. You have the lowlands, which will perform better in this blue region down here, and then you have in the middle, you have this transition zone in which both the lowland and the upland ecotypes will perform relatively well in this area. And that's actually where both the Rutgers and the Pennsylvania site lie, and the Cornell site kind of lies like right on the border of this. It's probably in the transition zone, but it's like right on the edge between the northern upland zone and the transition zone. So next, the phenotypic data used in this presentation, this was collected during the summer of 2019 and was the first of 3 years of data to be collected for this project. Measurements such as height, circumference, and anthracnose disease ratings were taken. Lastly, genotype analysis was done using Tassel version 5, and a mixed linear model was performed to make associations between the specific S&P markers and phenotypic traits. So now we're going to zoom in on our anthracnose results shown right here. So on the upper right portion of the screen, we can see a frequency distribution for anthracnose disease ratings at all 3 field sites. The Rutgers field site is shown by the red bars, Penn State corresponds to the blue bars, and Cornell corresponds to the green bars. Anthracnose disease ratings were measured on a 1 to 5 scale, with 1 being a low disease severity and 5 being a high disease severity. The Rutgers data was initially collected on a 1 to 10 scale and was then subsequently transformed to match the data collected from the other 2 locations. So the immediate takeaway from this histogram is that the distribution is skewed towards the higher ratings, indicating that there is a relatively high degree of disease severity across all 3 locations. Also, as indicated by the left side of the graph, shown right here, there is only a small fraction of plants with a low disease severity of a rating of 1. These plants, however, are actually the ones of greatest interest to us for use in breeding efforts, as they'll be the ones that are most resistant to anthracnose disease. So now we're going to look over here on the bottom left at this Manhattan plot. For this diagram, the S&P presence threshold was set at 25%. This basically, so out of the initial 700,000 S&Ps that were genotyped, approximately about 90,000 of them were retained. The significant threshold is depicted by this solid black line running across the diagram. It was set for p-values that were less than 5 times 10 to the minus 8. That corresponds to about 7.3 on this figure, as this figure on the y-axis actually is a minus log 10 of the p-value. So in total, this Manhattan plot shows that there was 19 QTLs that were found to be significant. And if we take a look at the QQ plot on the right, it shows that there's a divergence between the expected p-values and the observed p-values. And this is indicating that there's likely some type of population stratification and differences in allele frequency in this underlying population structure. So the next part that we're going to zoom in is on the results on the height and circumference. So both the height and the circumference histogram shown here on the left, they both show a roughly normal distribution for each of the 3 locations that were measured. The height measurements were ranging from relatively small plants that were less than 80 centimeters in height to exceptionally tall plants that were more than 200 centimeters in height. The Cornell and Rutgers distributions were similar for height measurements and were much taller on average than the Penn State location. The frequency of circumference measurements was similar when compared across all 3 locations and ranged from less than 10 centimeters to more than 100 centimeters. So using the height and volume measurements and making the assumption that the plants formed a cylinder when being measured, the volume of biomass could be estimated using the formula for the volume of a cylinder, which is equal to pi times r squared times the radius squared times the height. This is shown in the histogram over here on the right. So similarly what we saw with the height measurements, the Cornell and Rutgers locations, they averaged about 11,000 cubic centimeters, whereas the Penn State location had a volume of about 4,500 cubic centimeters. And some of the plants in the Rutgers and Cornell sites were actually quite massive, and they had volumes of more than 100 cubic centimeters. So when you look at, or when you compare the volume distribution to the height distribution, one thing should stand out is that they seem to be much more correlated together than the volume and the circumference. So this kind of points to a possible correlation that might exist between height and volume, as both the Rutgers and the Cornell sites were taller plants, and they also had higher volumes, whereas in their circumference measurements, all 3 field locations were roughly the same. So there might be a possible correlation between volume of biomass and the height of the plant. So the next part that we're going to zoom in on now is the GWAS for the volume characteristic. So here, the significance threshold was set to a p-value of 5 times 10 to the minus 8, or p-values that were less than 5 times 10 to the minus 8, which was similar to the Anthracnose study. However, this graph, the S&P presence threshold was set to 50%, which was resulting in about 29,000 S&Ps being retained out of the initial 700,000. A total of 8 QTLs were identified as significant, being above the line shown up here. And looking at this QQ plot for the volume, just as we saw with Anthracnose, there is a divergence between the expected p-values and the observed p-values. This is indicating that there's likely, again, some type of population stratification that's confounding. That's a confounding factor. So now we're just going to wrap things up by zooming in on our discussion section shown over here. So the QTLs that were identified in this first year of data will serve as a good starting point for the next 2 years of data that will be collected. We can look to see if there's any consistency between the QTLs identified between the years. We can also run individual genome-wide association studies for each of the individual sites, as opposed to running the combined data to see if we can identify the same QTLs in each of the locations. When when looking at those separately, this GWAS project is currently being done in parallel with the evaluation of breeding populations for high disease resistance and biomass production. As I mentioned earlier in the presentation, there are a few exceptionally large and disease-resistant plants within this association panel, and they may serve as a useful tool for making crosses with some of the germplasm that's already present within our breeding populations. Currently, we're currently in the process of constructing a genetic linkage map using a mapping population of switchgrass that's segregating for anthracnose disease resistance. This mapping population will be useful in identifying QTLs for anthracnose disease resistance. The QTLs obtained from this mapping population can then be compared with those obtained from the association panel, shown in this study, to further narrow down any potential underlying candidate genes for resistance. We can do this by using gene ontology, and we can look for potential candidate genes within these significant QTLs, and we can look to further validate and narrow down our search. We can look for QTLs that have been consistently showing up across years between the mapping population and the association panel, and this will give us a much better sense of which genes are actually involved with anthracnose resistance. So now I'm just going to zoom in briefly on the references I used to put this together. So I used these references to construct my background information and to help in aiding analyzing my results. And that is my presentation. I hope you enjoyed it. Thank you for listening.